Hi, everybody. Welcome to our session. On behalf of Yale University and Yale Cancer Center, I'm pleased to have you with us as part of the Yale Engage Cancer series. This session is entitled Defining Mechanisms and Biomarkers of Sensitivity and Resistance to Anti-Cancer Treatments. I'll be your moderator. I'm Barbara Burtness. I'm a medical oncologist and have an interest in drug development and head and neck cancer. And we have a phenomenal panel of Yale faculty members and a corporate guest, Susan Galbraith from AZ, and hope to have a very, very interactive session. I'd like to start with a few housekeeping items. The program format, as I said, is going to be each of our panel members giving a brief, about 5-minute introduction to the work that they do, what they see as key questions. We'll have all of the panel presentations first and then move on to the discussion, the question and answer. We know that to attack cancer, we need team science, we need collaborations within our organization and across different sectors, academic, public, and industry. And Yale Engage was designed to build these connections, particularly between Yale scientists and industry leaders. To keep the discussion lively, we welcome questions. Some have been submitted ahead of time, and you'll have the ability to submit them through the Q&A function on the webinar. We have an enormous amount of expertise among our panelists, and we'll be monitoring those questions as they come up and try to get to as many of them as possible. And I want you to know that this webinar is being recorded. So now I'm really pleased to be able to introduce Charlie Fuchs. He's the Sackler Professor of Medicine and Medical Oncology and a Professor of Chronic Disease Epidemiology here at Yale. He's the Director of the Yale Cancer Center and Physician-in-Chief at Smilow Cancer Hospital. Charlie?
Barbara, thank you, and thank you for your leadership on this, and welcome to all the attendees to what is now our third Yale Engage Cancer event. And it's really been an exciting and incredibly productive series of forums. So pleased that you could join us for this third one. You know, we all recognize that despite the fact that we're dealing with a global pandemic, the consistent impact of cancer on public health and the morbidity and mortality and costs on our population are considerable, and it remains one of the great challenges in medicine, and it also is one of the largest investments, I think, that goes on in healthcare research and drug development. And our efforts at Yale is to really tackle this challenge. Yale has had a long legacy in cancer research and cell biology, genetics, pharmacology, immunology, among other elements. And I think a lot of the history of success, including in chemotherapy, has come out of this university. We're privileged to work at one of the original National Cancer Institute designated cancer centers, and has been really an area that research that has built a legacy of great innovation. As well, Smilow Cancer Hospital, our clinical center, is celebrating its 10th anniversary and is a robust operation that now sees about 48% of every newly diagnosed cancer patient in the state of Connecticut. And really, we view that through the science and through this robust clinical operation, we really are committed to moving discovery, scientific discovery, into the clinic. Really pleased with the team that's been assembled today. Our first Yale Engage Cancer was focused on immunobiology. Our second was focused on novel therapeutics. And the third really ties it all together, which is to understand now, given these efforts to develop new drugs, new targets, how do we understand resistance? How do we understand sensitivity? And how do we further enhance our approaches to cancer therapy? Integral to this fight is our collaboration with industry. And we're so pleased to have Dr. Susan Galbraith join us as our industry partner on the panel. And we realize that so many of you in the audience come from the biotech and pharma. And really, part of this effort, beyond hearing from these experts and their insights, is to really begin a conversation. Because one thing we really welcome here at Yale is to collaborate with you. We want to build strategic partnerships with all of you. Because ultimately, this fight against cancer, yes, it requires each of these domains on the slide, but it requires a community focused on every aspect, and that includes academia and industry and biotech. So one thing I want to invite you today is to ask questions, but after this forum, please reach out to us. We'll reach out to you. And let's think about ways we can work together. I think we have a lot of resources we can bring here at Yale to partner with all the great things you're all doing. And we look forward to continuing this conversation long after this forum. So again, thank you for joining, and I'll turn it back to Barbara.
Thank you, Charlie. I think that's a great introduction to what we're trying to do here. I just had a brief opportunity to scroll through the list of 100 participants, and it's a formidable group, including Yale scientists, industry partners, alumni, our supporters. So I think that we can anticipate some pretty hard-hitting questions from that group. So we've tried to arrange these talks so that we hope that there's a little bit of a natural progression in the scientific questions and the approaches that are taken to understanding resistance. As I said, every speaker has been asked to sort of reflect a little bit on what's his core expertise, what questions drive the research, and how they hope to, or Yale hopes to, work with industry partners to address cancer treatment resistance, and what kinds of capabilities and resources need to be brought to bear. So each of the speakers has been asked to go only for about 5 minutes. I've been told that I should be ruthless and cut you off if you go over, and that'll be hard to do because I know the talks are going to be great. But let me start by introducing Dr. Katerina Politi. She's an associate professor of pathology and medicine, a leader in our cancer center through the cancer signaling networks program, as well as a leader of our lung spore program. And Katie, take it away.
Thank you very much, Barbara. And I'm really delighted to have the opportunity to speak here today and tell you about some of the things that we're interested in. I have a cancer biology lab here, really with a focus on lung cancer. And one of the areas that we are really interested in studying is working on resistance to various cancer therapies, including targeted therapies and also immunotherapies. And some of the things that we think about a lot and work on really approaches to discover new mechanisms of resistance. We're interested in understanding the relationship between tumor genotype and drug sensitivity. We study the influence of the tumor microenvironment on sensitivity to different therapies, and also investigate mechanisms of drug tolerance. So why do some cells die when you apply a therapy, and instead other cells do not die and stick around and serve as the fertile ground for the emergence of drug resistance? And then we also investigate new approaches based on the science that we discover to overcome and or to prevent the emergence of drug resistance. And we do these studies by really integrating information from various different systems, various different models, and using a variety of different technologies. We use specimens and data from patients. So we have a very robust biopsy program here within the context of the lung cancer group, where we can obtain biopsies from patients along sort of the spectrum of their treatment with therapies. And we can generate patient-derived models from these biopsies, but also then analyze the data and information to really understand resistance in patients. We use these models to generate, or these specimens to generate patient-derived xenografts as well, and also 2D or 3D cultures from patient specimens. And we also extensively work with genetically engineered mouse models of lung cancer that we can use to study resistance. And in that regard, I'd like to tell you today about some work that we have been doing in the field of EGF receptor mutant lung cancer. Next slide, please. To really use models to study resistance to the EGFR tyrosine kinase inhibitor Osimertinib. And this is work that really illustrates a partnership between academia and investigators in academia, and work that we've done together with AstraZeneca, and also working with Guardant Technology, and work that was published recently this year. And so EGF receptor mutations are found in about 15% of lung cancers, and can be targeted with tyrosine kinase inhibitors. And one of the most recent ones is this tyrosine kinase inhibitor Osimertinib. And so we can take our genetically engineered mouse models and ask the question, what happens if you have mouse models of EGF receptor mutant lung cancer and you treat them with Osimertinib? And so we took mice, we treated them till the emergence of resistance. And when we looked at resistant tumors, to see what was happening, we found that almost 50% of the tumors that emerged had secondary mutations in EGF receptor that conferred resistance to Osimertinib. And so with that information, we can actually then go ahead using these models. So we've discovered new mechanisms. We can now use these models for preclinical testing and test new therapies. We can also, with this information, go into human specimens and data and analyze the relevance of the resistance mechanisms there. And so, for example, in this study, we found that the mutations that were emerging were particularly relevant to the L858R subset of EGFR mutant tumors. So there was an allele specificity that was revealed through our studies in mouse models. And then working with colleagues like Mark Lemmon here, who you're going to hear from next, we can really then study the biochemical properties in detail of these mutants. Next slide, please. So we also are working extensively to take these models that we have and sort of take them to the next level to study some of the more complex mechanisms of resistance. And we have modified, for example, this initial mouse model of EGF receptor mutant lung cancer to incorporate additional genetic alterations that are also found in humans in EGFR mutant lung cancer, including, for example, tumor suppressor gene alterations using in vivo CRISPR-Cas9 gene editing. And so now we can study how those additional alterations are impacting tumor progression, sensitivity to therapies, and the phenotypes of tumors. As I mentioned in my first slide, we also have a robust program to generate patient-derived models. And here's really an illustration of sort of the different PDXs that we've generated across various different oncogenic subgroups of lung cancer with different oncogenic driver alterations. And so we're using these models to really study resistance in human specimens and really use them to study heterogeneity of human tumors, signaling network alterations, and the molecular profiles that you can have in these human tumors with or without drug treatment. Thank you.
Thank you so much, Katie. I think that there's so much there for the other speakers to riff off of and to set up our questions. Next, let me introduce Mark Lemmon, Distinguished Professor of Pharmacology. You see his leadership roles in the Cancer Center and Cancer Biology Institute there. And Mark is unique in bringing a wealth of expertise in biology and structural biology to the very interface with drug development and disease-based research. And so looking forward to your comments, Mark.
Thank you very much, Barbara. And good afternoon. It's a great pleasure to be here. I look forward very much to hearing the discussion later on. As Barbara mentioned, I'm really a basic scientist interested in how molecules work. My core expertise really is in biochemistry and structural biology. The focus of most of our work is detailed understanding of how molecules and networks involved in oncogenic signaling actually do work, and doing that in atomic detail where we can and quantitatively. understanding how their properties are changed by oncogenic and resistance mutations, as Katie mentioned, work we're doing with her, and how we can then use that information to guide mechanistically driven personalized medicine, or put the biochemistry into personalized medicine, those kinds of thoughts. So our main focus in general is the class of receptors that Katie discussed, the growth factor receptors that have intracellular tyrosine kinase domains like EGF receptor. As you know, and as Katie described, these are key targets for cancer therapy, particularly in lung cancer. And it's clear in general in advancing approaches to controlling their behavior, cellular behavior with drugs, dealing with resistance, really requires us to understand the molecular mechanisms and understand them well enough that we can manipulate them in a predictable way, and also manipulate their complex cellular networks. I'll give a couple of examples of things that have been driving research in my lab recently. The first really relates to what Katie has been discussing at the level of both acquired resistance and primary resistance. And we've actually been working with Katie quite a bit to understand details of how secondary mutations in EGFR cause resistance, as she mentioned with the Osimertinib resistance mutations. And an additional key goal in that work is to use that understanding as it develops to decide when to use which inhibitor and how to come up with new and indeed repurposed inhibitors in resistance situations. Now, going back to other work in the lab, one of our recent foci has been to identify and target driver mutations in neuroblastoma, which is one of the most common pediatric cancers. And this is related work. We've been collaborating with the Children's Oncology Group on ALK, another receptor tyrosine kinase, a bit like EGFR. And sequencing ALK in tumors from 1,600 patients gave us a list with COG of ALK mutations that we analyzed biochemically, structurally, and through transformation, did a full workup on them, and showed from that that ALK, about 14% of neuroblastoma is ALK dependent. And we developed a computational model that you can see in the middle of the left-hand part of the slide, that with which we can predict which mutations are actionable, and we're still working on that, and refining that to identify ALK-dependent tumors in the clinic. But importantly, this quickly led us to understand that some variants are resistant to first-generation ALK inhibitors. Crizotinib does not work in neuroblastoma. And we also learned that the stable of first-generation ALK inhibitors were not that different from one another. And in PEDS in particular, one really has to be careful to which drug you choose for the trials, because there's a limited number of patients in pediatrics. So one wants to pick the right one. And overall, those considerations using the biochemistry channeled us to lorlatinib, which is now looking promising in neuroblastoma, overcomes much of the resistance, although of course we're now experiencing resistance that we're working on. And so I just want to illustrate that as a key approach, combining biochemistry and structural biology and computational aspects that we could use in principle for any receptor tyrosine kinase. So next slide, please. We're also very interested in a newer aspect of getting away from inhibiting receptors per se, as we tend to do, and instead correcting their signaling. So we're all familiar with biased agonists for G protein coupled receptors, which can promote different responses through the same receptor, as illustrated on the left, the color of signaling, whether it's orange, yellow, green, or blue. Many common drugs that we take are biased GPCR agonists, and there's actually a lot of effort, for example, to develop biased agonists of opiate receptors, retaining the analgesic effects, but without the associated problems of the opiates. We don't do that for receptor tyrosine kinases and the like, because they're traditionally thought of as being binary signaling systems, either on or off, as I've illustrated here. But we recently showed in a paper a couple of years ago, which we continue to work on, that receptor tyrosine kinases have color in their signaling too. And as illustrated on the right, the same receptor, EGF receptor again in this case, can promote cell proliferation or differentiation, depending in the same cell, depending on which growth factor is used to activate it. And this reflects, we now know, a different dimer structure, asymmetric or symmetric for the two ligands, with altered dimerization and signaling kinetics that define specificity. It turns out the mutations in glioblastoma shift signaling to the right, making it more proliferative. That's one of their key issues, even with small structural changes. Now that we understand the structural basis for this through crystallography and so forth, we believe that it's possible to develop biologics that will do the opposite. Imagine, for example, an antibody that could shift EGF activated in cancer, or mutationally or with ligand, shift signaling to the left, making it differentiative, This could be a really powerful approach to signaling, switching or correcting signaling from proliferation to differentiation. There's actually one proof of principle in that with KIT and stem cell factor that Garcia's lab has been working on at Stanford. So next slide, please. And so finally, we've also been focusing on an undruggable target, the pseudokinases. About 10% of the kinome is inactive, and the blue ones here on the left in this tree. Many of them don't even bind ATP, and these include regions in receptors like RORS, RIC, PDK7 involved in Wnt signaling and involved in several cancers, but have been totally ignored as drug targets for the most part. One hypothesis is that they signal by switching conformations to bind downstream signaling molecules. We recently determined in this paper in 2020, it's referenced here, a bunch of structures and screened for small molecule inhibitors to see if we could, in principle, drug these. In the middle here, in the structure, you can see a drug. It's actually ponatinib, an ABL inhibitor, bound to one of these pseudokinases that doesn't even bind ATP. And then, as shown top right, we've demonstrated using hydrogen deuterium exchange studies that ponatinib induces conformational changes in ROR1 as it binds. And so the idea is that this might inhibit signaling interactions. Naturally, there's enormous amount of work to do with selectivity and so forth, but early studies of signaling effects suggest that ponatinib can inhibit Wnt-dependent ROR1 signaling. And so the idea shown centrally here is that conformational disruptors like this could be valuable tools for understanding pseudokinase signaling, but also targeting them where they play known roles in cancer and other diseases. And so far, they've all been hit. The RTK ones, for example, with antibodies. So that's about my brief summary. That's all I wanted to say. So thank you very much for your attention, and I look forward to your questions.
Thank you very much, Mark, for that wonderful discussion. Next, I'll be introducing Megan King, Associate Professor of Cell Biology and Molecular, Cellular, and Developmental Biology, a program leader in our Cancer Center, and I think notable partly for having been elected by her fellow faculty here at Yale School of Medicine as past president of our Faculty Senate equivalent, the Faculty Advisory Council, where she also showed exceptional leadership sort of in that other realm. And she's going to be talking to us about very impactful work regarding resistance to PARP inhibition.
All right, so thank you. I'm also a basic scientist, and over the past decade, it's really been my interactions with my colleagues here in the Yale Cancer Center that has driven my group with expertise in genome integrity to really focus on those aspects that have impacts for cancer therapies. So I want to start with this classic example of synthetic lethality, and that is specifically PARP inhibitors in the context of BRCA1 and BRCA2 mutations. And although, of course, these therapies have incredible promise, it's well established now that the acquired resistance is a major bottleneck for the durability and efficacy of these treatments. And so how do we tackle this problem, and are there opportunities that are presented when these tumor cells become resistant? And so the approach that we've been taking is first to start by really trying to define the genetic basis of resistance in this context. And so we know that there has been real value in CRISPR screens, but I think increasingly we're very excited about the possibility of circulating tumor DNA sequencing, as well as a potential for serial biopsies, particularly along this axis as tumors gain resistance, to combine with genome sequencing as well as gene expression analysis to provide new insights into therapy resistance. And we use a range of models, all the way from model organisms to mouse models, to really get at the mechanisms. And of course, the ultimate goal is always to really be able to leverage the mechanism of resistance, ideally to come up with new therapies. And so, while of course we'd like these to be actionable, we're really particularly would like to go beyond that and to be sure to consider, based on our mechanistic studies, what can we bring to the table in terms of stratification. And today I'll talk about an example where we really think that we have to consider BRCA1 patients separately from BRCA2 patients. Of course, it would be best really if we can develop new biomarkers that will further help us stratify patients based on the mechanisms underlying resistance. And I think one real potential there is, for example, circulating tumor DNA may allow us to identify patients who have a so-called reversion allele that now will make them insensitive to PARP inhibitor therapy. And that may be one cohort, but there may be other patients where resistance is arising through a secondary mechanism that may be therapeutically actionable. And so I just want to take you through the work that we've been doing, and not just my lab, but across our team to look at the genetic basis of resistance. So much of, again, these CRISPR screens have been published. The work that's been going on here at Yale really has taken advantage of a partnership that we already have between AstraZeneca and our team, particularly Ryan Jensen. And Ryan has been modeling reversion alleles that are arising from patient-derived ctDNA sequencing and testing really, is there still an actionable approach that we could use in these contexts or not by functionally characterizing the reversion alleles? What I'm particularly excited about at the moment is that Pat LaRusso has been leading a trial along with Kurt Schalper, who you'll hear from in a moment, where she has been acquiring these serial biopsies along this progression to relapse. And this allows us now to go in and really look not just at genome changes, but gene expression changes. And so these sequencing is ongoing at the moment, and we're really excited about the new targets that it may reveal. So I just want to give you one vignette of what was really originally motivated by these in vitro screens and some work that my group has done and the possibilities that we can see for this going forward. So it's well established that BRCA1, one of its key roles is to promote what's called double-strand break and resection through the exo-1 pathway. And this is a critical step in the HR pathway. And so it came out of these screens that loss of either 50-BP1 or Rev7 can drive therapy resistance in the context of BRCA1 mutations. What my group discovered is that these are negative regulators of the Bloom helicase acting with DNA2, which is an alternative end resection mechanism. So this is a way where these tumor cells have essentially rewired resection. So they're no longer dependent on BRCA1 and instead can use this Bloom pathway. And so as examples of what that mechanism has brought about in terms of the way we're thinking about future therapeutics, the first is that it identifies the Bloom helicase as a really novel target that we have already shown in vitro is also synthetic lethal with BRCA1 on its own. Particularly if we think in the short term, maybe more actionable in ways in which this has changed our thinking is that it highlights also the potential for combinations of PARP inhibitors and ATR inhibitors. And that's because the other thing we discovered is that this Bloom-mediated helicase is driving resection at very high rates. And this leads not just to functional resection to do repair, it actually leads to hyperresection. And ATR is an important negative regulator of resection. And so we think that this combination of treatments will push this hyperresection even further. And this is a really good rationale for why initially patients with BRCA1 mutations may not respond well to a combination with an ATR inhibitor. But when there is a mechanism that downregulates these particular proteins, that will make these tumors very sensitive to the combination. And so along those lines, we're currently just submitting an LOI with Pat LaRusso, where we are proposing to do a trial specifically in BRCA1 patients, because this is not a mechanism that's relevant for the BRCA2 patients, and hoping to really test this idea clinically. So thank you, and I look forward to the questions. I also just like to highlight that much of this work, as I mentioned, was a collaboration with AstraZeneca and is also supported very generously by the Gray Foundation.
Thank you, Megan. That was terrific. Next, I'd like to introduce Qin Yan. He's an associate professor of pathology and director of our epigenetics program here at Yale. And we'll be talking about epigenetic mechanisms of resistance.
Thank you, Barbara. My expertise is mainly on cancer epigenetics. And as you all know, epigenetic mechanism is critical for cancer initiation and progression. Especially my laboratory is interested in understanding how epigenetic regulators, or so-called reader, writer, and erasers of DNA methylation, histone modification, how they regulate different steps of cancer progression. My laboratory are interested in a couple of different areas. One is the resistant mechanism to anti-cancer drugs, which is the main topic today, cancer metastasis, and the tumor immuno-evasion, which is one of the areas that Kurt Schalper will talk more about later on. And next. My laboratory is also very interested in developing different epigenetic drugs, and we have done some work with Yale Center for Molecular Discovery, which is our in-house screening center. And I have done some work with the NCI Experimental Therapeutics program. And right now, I'm also collaborating with some biotech and pharmaceutical company in this area as well. And in the next 2 slides, I'm going to tell you some of the examples that we have done to look at the resistant mechanisms. Next, please. One of which is targeted therapy, and in this case, the trastuzumab, or called Herceptin, for breast cancer. And we can generate those resistant cells in tissue culture, and we found that those resistant cells actually do not have genetic mutations. The resistant mechanism is actually reversible. If you take the drug away from the cells for a short period of time, and they still maintain resistance. But if you take it away for a long period of time, for example, about a month, and those cells becomes those so-called washout, and those cells become sensitive to trastuzumab again. To look into the mechanism, next please. We profile the expression profile of the resistant cells compared to the sensitive cells. We can see that those resistant cells have increased oxidative phosphorylation, or called OXPHOS. And remarkably, those cells are very sensitive to OXPHOS inhibitor. As you can see, the tumor regression, if you combine trastuzumab and oligomycin A, which is one of the OXPHOS inhibitor, you can see regression of those resistant tumors. Next, please. As I mentioned, that there's an epigenetic mechanism that contributes to resistance. So one of the mechanisms we found is that KDM5 histone demethylase are critical for this formation of those resistant cells. We can combine with the targeted therapy and the KDM5 inhibitor, which this is one of the early generation inhibitor, and for to prevent the formation of the resistant cells for both breast cancer, which is BT474 cells, and non-cancer cells and PC9 cells. Next, please. So we are also very interested in understanding how resistance happens to immune checkpoint blockade. And this is our version of the cancer immunity cycle. And as you can see, there's actually 2 steps that are critical for immuno checkpoint to work: is the trafficking and the infiltration of the immune cells to the tumor. And apparently, some of the epigenetic modulators have been shown to be critical for those processes. And I will just show an example in our laboratory. Next, please. Where we found the KDM5B, or histone demethylase 5B, is critical for the infiltration and trafficking of the T cells to the tumors. And if we knock out KDM5B in those YAMA 1.7 cells, which is a mouse model generated by Marcus Bosenberg at Yale, we can see that if you knock out KDM5B, those cells are unable to form tumors. And if we re-challenge those mice with control cells, which normally grow very well, you can see they cannot grow, and meaning that those mice have gained immunity against those YAMA cells. If you look at the poorly immunogenic YAM 1.0 cells down in the panel C, you can see those cells are not responsive to PD-1 blockade at all. And if we delete KDM5B from those cells, you can see the slowdown of the growth of the cells. And if you combine with the PD-1 blockade, you can significantly extend the lifespan of those tumor-bearing mice. So this suggests that KDM5B is a very good target to overcome resistance to immune checkpoint blockade. And I just want to mention that this is done in collaboration with multiple labs at Yale, including Akiko Iwasaki and Max Bosenberg's lab. So team science is one of the same at Yale. We work together a lot. Thank you.
Thank you. That is such a terrific story. Now I'm pleased to introduce Kurt Schalper. He's an assistant professor of pathology and medicine and a recent recipient of an NCI Merit Award. He conducts really cutting edge immunoprofiling studies And look forward to your talk, Kurt.
Thank you, Barbara. So next slide, please. So I trained clinically in molecular diagnostics, and I've been working in cancer immunology for about 10 years now. And it's unquestionable that immuno-oncology has revolutionized the treatment of cancer, but there are major challenges still to overcome. So I'll cover a few of the challenges that I think are critical to potentially move the field forward. One of which is that I think there have been conceptual limitations of in drug development and identification of biomarkers. Relative to drug development, I think the focus of many people developing targets has been on immunostimulation, but that doesn't necessarily consider correcting alterations in the tumor. And this is critical because if we're only stimulating T cells, and there is not a clear gradient towards activating it more in the tumor, it's likely that the therapeutic index is smaller and the potential of benefit and toxicity balance is affected. So I think the concept is that we shouldn't focus only on stimulating T cells everywhere. We should probably look for signals that have a gradient favoring the tumor. And relative to the development of biomarkers for resistance, I think there have been a little bit of confusion in the field because immunotherapy has been used so widely that people are calling every patient that don't respond as a resistance. And conceptually, I think that's probably not accurate because patients without PD-L1 expression, tumor mutation burden, or any biology should not respond to start with. So I think there is a confusion between any patient that lacks benefit versus true resistance, which in my opinion are the patients that should have responded but didn't. I think this is critical to design programs and biomarker plans. The second important concept that it's connected with the previous one is that it's probably necessary to identify dominant immune evasion pathways that are well represented in the tumor. And this is for the same reason, because we need to have this gradient and strong biology in the tumor to be able to achieve a meaningful anti-cancer response. And then another major need in the field is try to identify potential targets that are beyond the T cells, so to have complementary effort and not have only redundant mechanisms. Another important observation is that we as pathologists, when we look at the tumors, we realize how difficult and how complex is the tumor microenvironment, where most interactions between tumor and immune cells are happening. And I think deciphering the tumor microenvironment and how different it is across tumors and across patients, it's a major need to really drive better biomarkers and better immunotherapy. Then also, I think we need to do a better work at understanding the interactions between major dominant oncogenic signals and immune evasion pathways. This has been somehow been revealed in EGFR mutant tumors that are less sensitive and less inflamed, but I think there's a whole world to discover what alterations in the tumor, somatic alterations, are able to manipulate immune response. And then finally, I think there are limitations of traditional studies, as we just saw from Jane. Many alterations are not at the genomic level, which is the favorite way we use to analyze the tumor side of the interaction. So I think by just doing genomic analysis, we're missing a lot of alterations at the immune system. And this, I think it's something we can overcome. And finally, I think that most of the studies are focusing on both ends, on the very early discovery type of work with CRISPR screens and other strategies. And then there is a huge effort on the clinical development. But I think there is room to improve some studies in more sort of human, real context. Next slide, please. So this is an example of the approach that we have taken in my group, where we generate hypotheses using discovery and biology, and then we actually have generated assays to screen for pathways, cell types, and tumor cell indicators in the same tissue. So we can actually do both genomic analysis to understand the genomic contexture and drivers, but then we can also look at the immune contexture and pathways that are potentially actionable. We have become pretty good at looking at multiple high throughput methods to detect protein level. And then we can do single cell analysis, spatial analysis, and really try to understand the tumor microenvironment. To prioritize what signals are dominant or relevant, we usually use regressive analysis using outcomes and response to treatment. So that way we can identify which signals are relevant from the ones that are not. Next slide, please. This is important because ultimately those signals are the ones we can validate in vitro to demonstrate that these are not just epiphenomenons or correlations, but they are mechanistically relevant. And then ultimately we can go back and look at this in the context of human clinical trials. And I'll show you an example of that. Next slide, please. So just to illustrate how this cycle works, this is a story that it's published this year where we identify interleukin-8 and local neutrophils in the tumor microenvironment as dominant immune evasion pathway and resistant mechanism. So the story started a few years ago where we look at in situ RNA expression for interleukin-8, and we found that it was producing tumor cells and highly associated with resistance to immune checkpoint blockers. So to advance this further, we look at the relationship between interleukin-8 and neutrophils, as shown in the upper side of the slide. And then we found a fraction of tumors that had upregulation of interleukin-8 and an unfavorable microenvironment characterized by increased TILs and fewer T cells. We also did genomic analysis to understand that this was independent from tumor mutational burden and major genomic alterations. And then we finally were able to demonstrate that the production of interleukin in the tumor was actually associated with interleukin-8 in serum, in circulation. So with that, we conducted a study in over 1,200 cancer patients from 3 phase 3 pivotal trials using immune checkpoint blockers. And we found that about a third of patients across tumors have upregulation of interleukin-8, and they have low sensitivity to immune checkpoint blockers. Next slide, please. Then to further demonstrate this, we did another study in which we cultured neutrophils and myeloid-derived suppressor cells to show the mechanism behind, and we were able to demonstrate that formation of nets was involved in effector response suppression. And then ultimately, we're working with a clinical trial where patients now are being treated with an antibody and targeting interleukin-8, and to understand if this pathway can actually be actioned in real patients. And hopefully we can use the biology that we figure out to drive the biomarker plan. Next slide, please. So finally, we have gotten a little bit more sophisticated now in generated models for in vitro tumor treatment. This is just an example of what we're doing, where we can culture primary tumors, treat them in vitro, but intact, so that we can then generate preparations and analyze the tumor microenvironment change, now perturbing these tumors with immunostimulatory or other anti-cancer agents. And we're incorporating new technologies such as single-cell transcriptomics and other analysis to do more unbiased studies. Thank you.
Thank you, Kurt. I mean, I think probably everybody can see the incredible power of that approach. Well, we said at the outset, Yale Engage is focused on building bridges and collaboration with industry. And in each of these seminars, we've invited an industry partner to speak to us. And I'm really thrilled that today it's Susan Galbraith. She's a Senior Vice President and Head of Early Oncology R&D at AstraZeneca. She's been there about 10 years, and in the early development program there, brought 7 compounds into phase 3. The story with PARP inhibition, the 3rd generation EGFR inhibitor Osimertinib that our colleague Roy Herbst was involved in presenting very impactful adjuvant trial this year. MEK and MET inhibitors, selective estrogen receptor directed agents, really a phenomenal portfolio and a phenomenal track record of success. So Susan, we look forward to hearing your thoughts.
Thank you, Barbara, and it's a pleasure to be here with you. So just a bit of an introduction. I'm a clinical oncologist by training, MD, PhD, and I've been, as Barbara said, at AstraZeneca for 10 years. And before that, I was in the US with Bristol Myers Squibb, also in the early development group, and stayed there for about 9 years. Just going to the next slide, I want to talk a little bit to build on some of the thoughts we've got about, understanding resistance. And one of the challenges that we've got about understanding resistance is really having access to the samples that would enable us to understand the clinical resistance. So Katie Politi has already talked to you about some of the models that we can use preclinically to model resistance. One of the challenges we've got with those techniques, though, is that it doesn't always predict what the true prevalence of the resistance mechanisms is going to be in the clinical setting. So if you start off with a PC9 cell and you look at the mechanisms of resistance to that, you don't necessarily understand what the true prevalence of all the things are when patients are starting with their own set of wiring diagrams in their EGFR mutant lung cancer. The other challenge that you've got is tried for a number of years to actually get biopsies from patients at the time of progression in clinical trials. And whilst we've included that, typically has to be as an optional biopsy at that time of progression, we've actually had across the range of clinical trials, relatively few of those actually materialize. And so that means that our mechanisms of understanding resistance during the development of osimertinib, have been somewhat limited. We started right at the beginning by looking at circulating tumor DNA right from the phase 1 trials with osimertinib. And we have some understanding and have actually published some of the data from the first line study with osimertinib, the FLORA trial, looking at those ctDNA mechanisms. But really, actually, one of the things that comes out of that is we could only explain just over a third of the patient's resistance mechanisms through looking at ctDNA. And the patterns that we saw there was we saw obviously the emergence of the cysteine 797S mutation, MET amplification, PI3 kinase pathway mutation and activation, P10 loss in some cases, and MET pathway activation as well in some cases. But really, the majority of patients, we still had a question mark over what the resistance mechanisms were. And so that led us to design this kind of study, what's called the Orchard platform study. This takes patients that are progressing on first-line osimertinib, and it offers them something that is potentially of benefit to them, which is to take a biopsy, to look at what the data says on next generation sequencing from that biopsy, and then to allocate them to a range of different potential arms. And there's biomarker matched arms, which you can see above, depending on the mechanism that is seen of resistance. And then there's also a non-biomarker matched arm. And this has been an important component of many platform trial designs, because it means that every patient who's given the consent to have a biopsy gets the offer of something. I can't guarantee that what they're getting offered is necessarily going to work, but it gives them that. And that has driven really quite a good uptake in terms of enrollment and accrual in this. And actually, one of the things that we've already learned now is we've now got data on over 60 patients, with tissue available at the time of progression in the Orchard study. And now that we have an identifiable resistance mechanism now in nearly 2/3 of patients, as opposed to just a third. We've increased the detection of some of the amplification mechanisms, which can be underestimated using ctDNA. We've increased the detection of some of the fusion mechanisms, which can also be difficult to detect using the ctDNA techniques. And we've got a better sense of the prevalence. There's still some work to be done here, and I still think we need to look at the epigenetic mechanisms that are driving resistance in this setting. But I just wanted to illustrate this as an example of one way that we need to look at in terms of understanding, documenting resistance and moving on from it. So if you go to the next slide, a similar approach has been taken in the understanding resistance to checkpoint inhibition. And I completely agree with Kurt Schalper's comment that not everybody who progresses on a checkpoint inhibitor is necessarily truly resistant, but I think we need to understand some of those mechanisms. And again, this is a mechanism where you can get the biopsies from these patients, also some peripheral blood sampling, and look at ways in which we can potentially offer them treatments that may have the opportunity to make a difference. So again, I just want to share with you a couple of observations from this. We go into the next slide. So first of all, there are some mechanisms that we might anticipate seeing based on really good data that's already emerged, and this is about the loss of heterozygosity for HLA or MHC. And we are seeing, as expected, that after treatment on one of these checkpoint inhibitors, there's an increased prevalence of loss of HLA or MHC, inability of the tumours to be seen by and be affected by the adaptive immune mechanisms if the antigen can't be presented effectively. Next slide. Other things that we're doing, you've seen a range of different mechanisms that we have looked at in this setting. we also mentioned the fact that obviously we're looking at the ATR combination with olaparib in terms of PARP resistance. But in fact, actually, one of the observations that we made early in phase 1 with our Osimertinib, which is our ATR inhibitor, is that we were seeing some unusual responses in patients that had a prior checkpoint inhibition in some of the trials. And so that led to some further investigations. So Osimertinib and durvalumab is one of the arms in the HUDSON study. And some of the data that we're seeing is quite interesting in seeing that you're getting a decrease in exhausted T cells, exhausted NK cells, and an increase in antigen presentation in patients that have both got primary resistance to checkpoint inhibition and subsequently have had some degree of response and subsequently progressed as well. And we're also seeing it not just in the ATM mutant patients that are selected, but also more broadly. So this is just an interesting observation. There's a lot more mechanistic data that is required and that will be followed up in order to understand this better. But I do think that these kinds of trials are really helpful in trying to understand the clinical prevalence of resistance mechanisms and get a lot more data that can feed back, back with the preclinical work that we can do to then understand what we might do next. So I'm going to stop there, and I'm very happy to address any questions that you might have. Thank you.
That was fabulous. Thank you very much. I am now going to ask that all of the panelists turn on their audio and video, and we'll now go into the full discussion. And I'm going to ask the attendees to please continue to post questions. We are monitoring these. And the first one, I think, basically immediately follows the last slide that we saw. And so maybe I'll ask Susan and Kurt both to address this. How critical is it to overcome the mechanical functional barriers to immune checkpoint inhibitors? And the questioner relates this specifically to HLA loss, although I can think of other mechanisms related to hypoxia and vascular alterations as well. But can you please comment on potential pathways and targets to overcome mechanical and functional barriers to immune checkpoint inhibitors? And Susan, do you want to go first and then kick it to Kurt?
Yeah, well, the cancer you think of, well, I think of first when you're talking about mechanical barriers potentially is pancreatic cancer, because of the high level of desmoplastic stroma that you see there, that has been discussed as not just having actually a physical potential barrier to treatment, but also the presence of the constituents of that desmoplastic stroma may also have biochemical effects that reduce the likelihood of sensitivity to of the tumor cells that are adjacent to that to treatment. And I think there are a lot of data suggesting that understanding the components of the microenvironment, the distribution and types of cancer-associated fibroblasts, for example, in that disease, and their phenotype might be absolutely critical to understanding mechanisms of resistance and sensitivity. I think in the context of loss of HLA, loss of HLA may increase the sensitivity potentially to other mechanisms, like inducing the innate immune system rather than the adaptive immune system, to NK cell enhancement potentially. So there are things that it then creates as a vulnerability, I suppose. I think the issue from my perspective is, you wouldn't be expecting, a high likelihood of subsequent response to something that requires HLA antigen presentation if you've got loss of HLA as a fundamental mechanism. So we should be segmenting patients by an understanding of these mechanisms in order to identify the populations that might best be subsequently treated with different kinds of therapies. Kurt, any thoughts from you?
Yeah, so I agree with all the comment. I think there's more biology emerging suggesting that the mechanical barriers may not be so mechanical. Some of these fibroblasts, they secrete inhibitory molecules, so it may be also an active immune inhibitory component to that. And that, I think, is thriving. I think that we're going to see a lot of new studies showing active mechanism of rejection of immune cells in the tumor bed. And relative to the antigen presentation, we have actually a study under review that should see the light soon, where we look at large cohorts of tumor mapping different parts of the antigen presentation pathway. In a long story short, what we've learned is that when we look at the genomics, we don't see that. So the majority of alterations are non-genomic, meaning non-mutation related. And the second interesting lesson is that depending on what molecule is lost in the tumor cell, meaning HLA-A, HLA-B, C, beta-2M, or other proteins, the immune contexture changes. So I think understanding that part will be critical to understand how to treat those patients. We do see upregulation of natural killer cells in certain loss of HLA molecules, but not in everyone. And each of them has sort of a certain different balance between T cells, NKs, and other cells. So I think it will be critical to do those studies to understand how granular this is. And if we can lump the antigen presentation defect into one category, or maybe it will be more than that. I think that's to be figured out.
So just continuing on with this theme and a question for Qin, can HLA loss be overcome by epigenetic modification, or what is the epigenetic role in HLA loss?
So this is not an area I have been working on very heavily, but as Kurt just mentioned, a lot of those changes are non-genetic changes. So we have different tools to activate those genes, reactivate those genes, and to make them successful, to make them to be sensitive to our treatment. So immune checkpoint blockade will work if you reactivate those.
Terrific, terrific. I have a question that was submitted earlier, but I think could probably be answered extensively or exhaustively by each one of the panelists, but maybe I'll ask Katie and Mark to start on this one. How does the mutational landscape of a tumor affect resistance and sensitivity? And I'm interpreting that the questioner means the other mutations besides the one in your target molecule.
Thank you. Sure, I can get started with that. I think this is really an area that we are starting to learn more about as we have learned more about the mutational profiles of tumors and of different genetic subgroups of tumors. So now one of the things that we've been able to look at, for example, are if we think about lung cancers in different oncogenic driver subgroups, we can look at the pattern of co-occurring genetic alterations that happen. So I'm thinking about, for example, in KRAS mutant lung cancers, these can co-occur with P53 mutations. They can co-occur, for example, with mutations in STK11, also known as LKB1. And we're really beginning to learn about what it means if the tumor has a KRAS mutation and a P53 mutation versus a KRAS mutation and an LKB1 mutation, for example, and that the LKB1 mutant tumors seem to have a different or reduced sensitivity to immunotherapy treatment, for example. And in parallel, I think similarly, with targeted therapies, we're really starting to scratch the surface and really beginning to start to understand how different co-occurring alterations also impact response to targeted therapies. So, for example, some of the work that we've been doing recently, looking at different tumor suppressor gene alterations in EGFR mutant lung cancer and how they affect sensitivity to tyrosine kinase inhibitors. One of the things that has emerged from our studies in animal models and also is emerging from studies of patient specimens is that if you have EGFR mutant tumors that also have mutations in the KEAP1 axis, so the KEAP1 NRF2 axis that is important for the antioxidant response of a tumor cell, if you have mutations that co-occur in that pathway, you have a decreased sensitivity to tyrosine kinase inhibitors. So the tumors will shrink less on treatment with these targeted therapies. And so that begs the question, is that a subset of patients who you could, for example, select initially for treatment with different therapies or for combination therapies together with a tyrosine kinase inhibitor, so that you could improve outcomes in patients with that disease. I think, of course, these types of landscapes, also this study in these landscapes really requires a lot of mechanistic investigation to understand exactly what is happening in those tumors. Finally, I think one of the other things to think about in terms of the genetic landscape also has to do with the overall mutation burden and the overall tumor mutation burden, which you know, we talk a lot about it in the context of immunotherapies, and where you know, we've heard about it, a lot about it in recent years. I'd say also there's some evidence that in the context of targeted therapies, the overall genetic landscape or the tumor mutation burden can have an effect on the response to targeted therapy. So again, in EGFR mutant lung cancer, tumors that are in the highest tertile of tumor mutation burden, which is generally lower than most other lung cancers, but in that highest tertile seem to do worse on treatment with targeted therapies, with tyrosine kinase inhibitors, than the ones with a lower tumor mutation burden. So there are lots of different aspects to consider, the specific mutations, so qualitatively, but also quantitatively.
Yeah, and I would just add to that, or add a kind of a broad conceptual thought to that, which is ultimately, I think, with all of these, with all of the therapies we're talking about, one is really trying to correct the signaling network, however you define network, whether it's intracellular, intra-tissue, intra-organismal, it's really a network. And in a sense, if you think about the fact that cancers are really caused by the networks losing robustness and kind of careering out of control to uncontrolled proliferation and so forth, it's almost surprising actually that a targeted therapy can work. And indeed, actually, if you create models where you've just mutated something we're hitting with a targeted therapeutic and nothing else, you don't actually, that's not enough to cause cancer. So the context is key. And the target that we're trying to correct is really just a kind of an Achilles heel in a sense for the rather plastic tumor in some senses. So I think that the answer to an answer to the question is that we need to think about these things as networks. We need to get into considering the systems biology of this. And I think there are two ways of thinking about that. One, and you'll all be aware of this, is the enormous effort put into machine learning, AI types of approaches, where as we collect more and more data for the mutational landscape to try to understand, you know, with various, you know, principal components analyses and what have you, how we can correlate combinations of mutations with sensitivity and so on and so forth. But there's another element that I think we have to consider that a variety of systems biologists are taking, which I think is really key. And actually, I think RAF inhibitor resistance illustrates this very nicely, is that we can actually learn an awful lot about how the networks operate. You know, a classic example is if you have a RAS mutation, then a RAF inhibitor does the wrong thing, you know. But so the bottom line is, I think that we need to start thinking beyond the targets to the networks and what the effect of the targeted therapeutics is on the networks. And that of course is going to hold in the immune context too, because again, what you're actually correcting, as Kurt and Susan have pointed out, what you're actually dealing with there is trying to restore balance in an incredibly complicated intercellular network. So I think that there's a couple of perspectives I would like to add.
Could you just add to that by introducing people a little bit to what's going on at the Systems Biology Institute? Not all of the audience may know about the scale of the effort.
Yeah, indeed. So actually, at Yale, we have a Systems Biology Institute that's actually headed up by Andrei Levchenko, who's the director of it. And Andrei is also a PI on one of the NCI Cancer Systems Biology centers. The NCI has a physical sciences and oncology center that actually initiative that actually I'm involved with one of those at Penn, and also the Cancer Systems Biology Centers Consortia that Andrei runs. And so the Systems Biology Institute here is really very cancer focused. It has a lot of interactions with the Cancer Biology Institute also on West Campus at Yale, and and Andre is an integral part of the Cancer Center. Of course, the Cancer Biology Institute is too, and so are most members of the Systems Biology Institute. And the kinds of things that are being looked at there, which is actually related to this, so that, Barbara, it's a good point, are, for example, Andre is very interested in looking at cells from brain tumors in particular, and asking and looking at migration versus proliferation in those cells, and the epigenetic difference between those in terms of what's defining the signaling networks that cause the cells to behave very differently. And he can do that with some microfabricated devices where you can separate cells based upon how rapidly they can migrate, and then go in and look with various single cell and other technologies to look at their transcriptome and obviously genome and epitranscriptome, etc. So that's really very exciting. There's another element which is really another two elements related to that. One is Sidi Chen, that many of you will come across. Sidi is doing an awful lot of work in terms of in the Systems Biology Institute too, in terms of trying to understand using in vivo CRISPR technologies origins of resistance to immunotherapies and other therapeutic approaches. And so that's really very exciting, and again is plugged into the network consideration of what's going on. And then one other aspect which I think is really cool, and actually is a project in our in the U54 that Andre runs, is that Gunther Wagner, who's an evolutionary biologist, is very interested in why certain mammals don't tend to get metastatic cancer. Cows in particular, as an example, the cows tend not to die of cancer. They carry them around. They carry the tumors around with them without metastasizing, and pretty much we don't see them here because they're all killed before they get to that stage for other purposes. And so that's fascinating. And again, it's looking at the network context, the network differences between the mammals that do and don't suffer from metastatic cancer. And actually, it's kind of related in some senses to placental invasiveness in these organisms, which kind of makes sense in some ways. And so there's a cost. There is a cost in some ways of having a placenta that more interdigitated, which is that you're more susceptible to metastasis in your cancer. So these different perspectives, whether it's immuno-oncology approaches, targeted therapeutics, I think there's a burgeoning, and it's very strong at Yale and really very exciting, I think, a burgeoning effort and understanding of how to put this into the quantitative, and I think it needs to be quantitative in the sense of biochemical networks and pathways contexts.
Wonderful. That was perfect. I have a question here asking the panel to comment on the role of proteomics in studying tumor resistance. And maybe you'll take that first, Mark, and then we'll kick it to Megan.
I can pick that. The perfect question. It relates exactly to what I was saying. I mean, as a biochemist, my view is that biochemistry is defined by the combination of components that you have, and a lot of those are proteins. Of course, it's not just proteins, and there are lots of other things too. But omics of various sorts are crucial for really getting a quantitative handle on understanding signaling itself, response to therapeutics, and of course, therefore, resistance. And I think there are two components of it. One is just, and I would just want to stress this, one is just the, I guess, fingerprinting approach that one often sees with proteomics, what proteins are there in a snapshot, what metabolites are there in a metabolomic sense in a snapshot. That's one aspect, and that can give you a lot of information. But looking at changes in the proteome, changes in the metabolome with time is really a crucial aspect, because really that's what gives us a picture of the networks that we're trying to correct and corral when we're targeting them with all of the therapeutics that we're discussing. And I just point out that there's quite a lot of activity on this at Yale, and one of the people recruited into the Cancer Biology Institute, for example, is Yansheng Liu, who has been doing a lot of work looking at, for example, proteomically both at snapshots and the time evolution of protein content as a consequence of aneuploidy. And, it's kind of interesting. You really, if you look at aneuploid cells, the effects on the proteome are really not what you would have predicted based on what you've lost in terms of gene copies or gain. And moreover, it's important to note that, and Rian Cheng has really pioneered this too, that RNA-seq data and proteomic data have substantial discrepancies. And so that means also that the proteomics is really an important thing to add to all of this. Kurt.
Yes, so I think I would like just to comment about the frustration of analyzing tissue level data without spatial resolution. This is critical because as Mark pointed, we see a striking difference between RNA and protein, which is becoming the rule more than the exception. But second, the protein measurements and any analyte really is context dependent. So for example, just to give you a rough example, a measurement of K67 protein in any given sample, if it's in the tumor cell, it means bad, tumors are proliferating. If it's in the immune cells, it means good, T cells are expanding. So really, I think having the possibility of looking at the proteins in the context of tissue organization, it's critical for understanding what's going on. And I think that's a little bit what I tried to reflect in my presentation. We're still early, we're getting more quantitative, the throughput is coming up to speed, but I think it's an important dimension of the protein and any analyte data.
Absolutely.
Perfect. And Megan, do you want to just jump in on this one as well?
I just wanted to add.
He does also. Okay, great.
First is to say, I think this is exactly right. This is why we know that just doing the genome sequencing is not going to be sufficient. We can start with the gene expression analysis because there are these epigenetic changes, but it's very clear that that's also not sufficient. And one thing I just wanted to point out is that as a cell biologist, I think we understand really well that, for example, translational capacity is something that's highly affected by stress. And so when we think about what's going on in a particular tumor environment, how that might be affecting the relative efficiencies of translation, which will never be really reflected in an RNA-seq data set, is going to be really important. And one of the things, for example, that we think about a lot because of Peter Glazer's work is thinking about hypoxia as a good example of this. And then the protein turnover aspects, right? And so these are all the factors that are contributing to what we might see different in a proteome data set. And I was going to make exactly the same point about aneuploidy because we know, and this is particularly relevant also for DNA repair factors, that one of the ideas of why aneuploidy causes such changes in the proteome is that we have these large protein complexes which are very codependent, and they become kind of out of titration with regard to the components. And that can, most of us are working on complex molecular machines where that's going to have an impact. And so that's going to really require detailed kind of mechanistic analysis. But we might get pointed to the fact that we need to do that work only if we go actually looking for proteome-wide data instead of just the genomics. Thanks.
Great. Katie?
Yes, so I just wanted to add to the conversation that because of the challenges of studying the pathways in patient specimens directly, all of the things that were brought up, the patient-derived models actually represent a really useful system to look at signaling and how signaling changes with treatment. And it's one of the reasons for which we did engage in this effort in developing these models, and also allows us to really explore how heterogeneous these samples are across different patient tumors. So we can take tumors with specific alterations or just across that were resistant to specific therapy, and we can look at specific things in terms of at the protein level in those, and we can look at if we apply other therapies, what changes. And so that really, I think, is a very valuable system in which to study what's happening at the protein level and signaling.
Susan, could I just ask you to comment on how the different omics are approached from within an organization like yours, and to what extent leveraging systems biology approaches is practical within your organization?
Yeah, sure. So I mean, I agree with the comments that have been made, first of all, that we do need to look at these different mechanisms, and it is possible to do that increasingly. We are looking with things like multiplex immunofluorescence at the spatial organization of the of tumors, and in doing that in patient samples now. So I think the technologies are advancing to enable you to do that. Single cell sequencing is also helping. I think, you know, what I would say is that you can't do that intensively on many trials. So you have to choose the trial setting and the context for that. And it does have to be complemented by the kinds of things that Katie has talked about as well. So I think, you know, you can see some sense of the overall picture emerging from some of the clinical trial data. You really need to understand the mechanism, and for that you need a different setting and an environment to do that, and the different techniques. And then, you know, the PDX models have some challenges. The GEM models have their own set of challenges. The humanized models for IO have their own set of challenges. And I think what we can try and do is, by looking collectively at you know, the clinical sample data and these range of preclinical models, and backwards and forwards across that divide, that's how you build up the bigger picture of understanding. And you know, I think it is like trying to sort of work out the overall picture from having several pieces of the jigsaw together, which is why you know, having that holistic view is absolutely critical to understanding. So you know, my comment would be that I think that the technology advances are now in place to enable us to see so much more than we were able to see 5 to 10 years ago. We need to bring that together, but have an integrated plan that goes across preclinical, translational, and the clinical trial environment. I think that's critical. So we spend a lot of time in what is called early stage oncology with our translational medicine group, actually working with the drugs and programs that are in late phase development or already on the market, like Osimertinib and you know, Developmental, and the other drugs that we have there, because for two reasons. One is that I think that's absolutely critical to understanding how to develop those and continue that. But secondly, that understanding then feeds back into the discovery organization for new opportunities. And I think the final piece I would say is that we can't do it all internally. Collaborations with organizations like Yale is absolutely critical. You've already heard a number of examples of the kinds of collaborations that we have that really help to feed and stimulate the work that we're doing internally. So I, you know, really appreciate the work that Megan and her team are doing, that Katie Politi and her team are doing, because that feeds what we're doing internally, and we can't do all of it.
Great, good. There's a question here asking, can we share examples of partnership with other academic centers that we may have for precision medicine efforts and adaptive combination treatment to overcome resistance? And I'll talk about that maybe a little bit from the medicine side. Pat LaRusso here leads Experimental Therapeutics Clinical Trials Network UM1 grant that has many consortium members, and she and her colleagues are leaders in taking molecularly driven questions and actually molecular selection strategies forward in the ETCTN network. Jeff Sklar here runs one of the labs that did the precision medicine sequencing for the MATCH trial. We have investigators here leading MATCH sub-trials. We have, I think, within our SPORES collaborations across other cancer centers that are molecularly driven clinical trial questions. So I think from the disease-based and clinical arena, and from the phase 1 arena, there is quite a rich network of these types of interactions. I don't know if anybody wants to address more from the preclinical level.
I could just comment that the anaplastic lymphoma kinase work that I mentioned, ALK work, that's all done as a long-term collaboration with people at Children's Hospital in Philadelphia and UPenn, where much of the computational modeling is done. And actually, through that, then another approach, another aspect that we're working on, which is a collaboration of many many academic medical centers actually in the US and abroad. We actually have, through the Alex's Lemmon Age Stand Foundation, an approach to targeting MYC signaling. Let me say, it's not targeting MYC per se, but relates to combination therapies and so forth. The idea being that so a multi-group approach with the idea that MYC aberrations, particularly MYC and neuroblastoma, affect the network, and that in principle one could rescue the network with appropriate combinations. And I think with the technologies, as Susan pointed out, advancing, I think the time is right to get to ask that question in that type of way.
Anybody else want to? Okay, there's a question here in the context of overcoming resistance. Can panelists share what they're most excited about in terms of combination modalities? And maybe I'll just ask Qin to go first, because I love the KDM5V story. But I think everybody probably has their own favorite combination. Qin, do you just want to?
Yeah, so of course, I think because I work on epigenetics, my opinion might be a little bit biased,
Sure.
but I think epigenetic modulation is critical for that. And not only so one of the example I have showed is the KDM5V, where we can show pretty synergistic effect of immune checkpoint blockade in multiple models. I only showed one. In breast cancer, we also see that as well. In addition, and there's other modalities that you can actually modulate the tumor microenvironment. And for example, some of the epigenetic regulators, as we are working on, one of them is called the CCR2. We can, by inhibiting that, we can change the macrophage population, and by doing that, we can change the T cell activity. So basically, it's just modulating the whole tumor microenvironment and make it sensitive for the immune checkpoint blockade. And this works well in a metastasis setting. So I'm quite excited about that. And as many of you probably know, in other institutions have started with DMT inhibitors, HDAC inhibitors, and EZH2 inhibitors, and those actually have showed strong efficacy in many different models. So I'm quite excited about this kind of combination.
Yeah, I think in Karen Anderson and Del Yarbrough have been working on demethylating therapy to uncover immune silencing in virally associated cancers. I think there are a lot of examples.
Barbara, can I comment on that?
Yeah, of course.
So I just wanted to comment that I think that's a very hard question, because I think ultimately the best combination is not going to be one combination that works every time. I think it's so clear now that the tumors evade immunity through different dominant pathways, and more advanced tumors tend to have multiple pathways, that I think the question has to do with where an immune evasion pathway is dominant, and where more than one is dominant. And I think that should drive the combination, not think the opposite way and think that one combination will fix tumors with different problems.
Yeah, so I think it's very interesting that you heard that question as an immune resistance question. It was a very broad resistance question, but that's an interesting perspective. I think one of the things that I think about sometimes is how some of the same mechanisms that have generated resistance to conventional therapies are now also generating resistance to immunotherapy, and how our relentless focus on target instead of environment, which I think is something I've heard you speak about a lot, Kurt. We may have the same Achilles heel over and over again. And in head and neck cancer, a clear example of this is hypoxia, which leads to resistance to DNA damaging agents. It leads to resistance to radiation therapy as the prime example, but is now increasingly linked to resistance to immunotherapy as well.
Yeah, and I'd say even taking it down to the simplest level of targeted therapeutics, the answer is it depends, because for example, just thinking about RAF inhibitor resistance, actually David Stern and Marcus Bosenberg and others did a nice study that they published a few years ago of combinations of drugs in a variety of cell lines from melanoma and elsewhere, and showed that which combinations work in which cells is very variable. And actually one of the things that I'm quite excited about at the moment, we're working as a group with Systems Biology Ireland and a group at Los Alamos and a group at Penn, trying to understand that in terms of the signaling networks around RAF and MEK and RAS in different cells from different cancers. And in a sense, which combinations work depends, say, on the level of KSR1. That's a key determinant. And so it just depends on so much on how the network is wired, which of course goes back to the question about proteomics, because ultimately you're trying to control the system, and the way the system is set up biochemically, it defines how it will respond to different combinations. And so I think we're going to want to get into things at that kind of level to understand when we should use which combination. And I think, to Kurt's point too, that's exactly the case is exactly going to be the case in the more complex systems of intercellular communication too.
And can I just add, there's also the kinetic component, right? So I think one thing we're still not clear on is, are you better off with this combination early on, or is this going to be better once you get initial resistance? And actually that may seem trivial, but I really don't think it is, and requires modeling to think about just how the kinetics is playing out.
Great.
So related to that, one of the things that I was going to say is one of the things I'm excited about sort of going forward and looking at the field over the next few years is really what we can learn about the tumor from the get-go that can tell us how we would want to treat it to stave off certain mechanisms of resistance. And I think we're starting to see some examples also of clinical trials that are starting to subset patients with certain tumor genotypes, or where the tumors have certain features, and sort of put them into trials with specific combinations. And I think that's going to be a really interesting approach in the next few years.
Terrific.
Thanks. Just a couple of comments on this. So there's a few things that you've heard there that I'd just like to build on. I completely agree that we need to understand what's the right combination for the setup of what the adaptive mechanisms have been in that particular tumor. And I think to that end, looking at the adaptation at an earlier point than we typically do is a key part of this strategy. So there's some really interesting concepts here that been working with Gordon Mills from OHSU on, and looking at the adaptive rewiring that goes on, which really happens quite quickly. And of course, one element of that we need to understand is undoubtedly the epigenetic mechanisms that come in, because they're quite commonly involved in some of the resistance mechanisms. And as I pointed out, we don't really have good techniques for looking for those if we haven't got a biopsy early on. So I think that's absolutely key. And then I think that also informs the potential for how we do combinations, because one of the limiting factors of actually getting these to work has been the tolerability of the combinations clinically. And for that, I think there was a couple of chinks of light of what we can do. First of all, we're starting to develop some better tolerated therapies inherently. So I think that gives us a bit more headroom for some of the combinations that we need to do. Things like ADCs, better ways of delivering some of the mechanisms of killing, give you a bit more headroom, and understanding what drives tolerability. And then looking at the sequencing rather than trying to dose everything at the same time is another innovation that I think will come in that will help us with that. But again, I think we're going to need to apply all of these tools that we've got and the modeling of that to reduce down the number of options that we actually bring into the clinic and increase the likelihood of each of one of those being successful. I think combination therapy is going to keep us occupied for a little while yet before we solve that problem.
Terrific. Great. Well, I hope the attendees have enjoyed this exchange of opinion and knowledge as much as I have. I'd like to turn it back now to Charlie Fuchs and just ask him to share a couple concluding remarks.
Well, Barbara, thank you and all the panelists. It was a fantastic discussion and really provided so much insight in terms of how we continue to move great science into the clinic, and frankly, how we learn more about the tests and clinical trials that we're actively doing as part of our investigation. This, as I mentioned, is the third of our Yale Engage Cancer Forums, and I hope that our attendees enjoyed it and benefited from it. And as I mentioned, the work continues, and we very much want this to be the beginning of the conversation. And so hopefully what you'll do and what we'll do is engage each other in thinking through how we partner, how we work strategically together to think of the ideas that our panelists have brought up and develop new initiatives. So people should feel free to reach out to me or any of the panelists to think about these collaborations. We'll be contacting you. Really appreciate you taking the time. You know, when I listen to discussions like this, I think it gives all of us hope and excitement about the years ahead of cancer investigation. So let me just turn it back to you, Barbara, for some final thoughts.
You know, so I've been at Yale about 6.5 years, and the conversation today sort of reminds me of the excitement that I felt when I started going to seminars around here. I mean, there's just a unbelievable scale of work of this quality going on at this institution, and a lot of people thinking about how to make cancer treatment better. So thank you all for joining us today. Please stay in touch. And I want to thank Susan and Kurt and Katie and Mark and Megan and Qin for their wonderful presentations. Thank you very much.
Thank you. Bye-bye.